DNA is a double helix. It's a double helical molecule where two polynucleotide strands twist around each other. See, there are two polynucleotide strands which are twisting around each other, and the polynucleotide strand strands have sugar phosphate backbone. This is the sugar phosphate backbone, and these are the base pair. Now, let us show what is happening here. As we have said, there are how many strands in DNA? Two strands, polynucleotide strands. And this is a single nucleotide. This is another nucleotide. And this is another nucleotide. And this is the 5' end, because phosphate which is attached at carbon 5 is free at this end. And the sugar which contains hydroxyl group at third carbon, this is also free here. So this is the 5' to 3' site. So this strand run in 5' to 3' direction. On the other hand, the complementary strand, polynucleotide strand, is running opposite direction. You can see here that 3' hydroxyl or carbon 3, third carbon of this sugar is free here, and the phosphate which is attached to the carbon 5 is free here. So this is running in 3' to 5' direction. So the two polynucleotide strands run opposite direction, So this is the sugar phosphate backbone, and this is also the sugar phosphate backbone of another strand, that is the complementary strand. And these are the nitrogen bases which are base pairing to each other. So how this base pairing occurs? This base pairing occurs by hydrogen bond. Thymine is always complementary to adenine. A thymine always links with adenine by 2 hydrogen bonds, or adenine links with thymine with 2 hydrogen bonds. But cytosine links with guanine, or guanine links with cytosine by how many hydrogen bonds? 3 hydrogen bonds. Okay?
Yes, sir.
So cytosine cannot link to the adenine, or adenine cannot link to the guanine, or thymine cannot link to the guanine, guanine cannot link to adenine. There should be purine pyrimidine base pairing, and for adenine there should be thymine, for thymine there should be adenine, for cytosine there should be guanine, for guanine there should be cytosine. So this is called a complementary base pairing. This is called what? Complementary base pairing. So DNA is a double-stranded molecule where two polynucleotides twist around each other. They have their sugar phosphate backbone, which are held together by this base pairing between the nitrogen base by hydrogen bonds. And RNA is a single nucleotide molecule, single polynucleotide, single polynucleotide stranded molecule. Okay, RNA is single stranded, but DNA is double stranded. Okay, there is no complementary strand in where? In RNA. So see, this is the famous DNA structure, model of DNA structure, double helix, model of DNA double helix made by Watson and Crick. And this lady is Rosalind Franklin, whose X-ray diffraction pattern gave important clues to the discovery of the model by Francis Crick and James Watson. So let's see what is the difference, basic difference between DNA and RNA. DNA consists of what? Deoxyribose sugar. So there is no hydroxyl group at second carbon of the sugar. That is why this is called deoxyribose sugar. But in RNA, there is ribose sugar and hydroxyl at carbon 2 is present. And you won't never find thymine in RNA. This is the uracil. The difference between uracil and thymine is that the thymine contains an extra methyl group which is not present in RNA. So there in DNA, the nitrogen base uh, is always thymine, but in RNA, no thymine, there will be uracil. Okay, see, this is the double-stranded DNA molecule where the nitrogen bases are cytosine, guanine, adenine, thymine. But in RNA, there is a single polynucleotide, and there is also cytosine, guanine, adenine, but no thymine, rather uracil. Okay,
Yes, sir.
so that's all about the structure, molecular structure of DNA and RNA. Now, let's see what is the structure of ATP. ATP, we know that ATP is adenosine triphosphate. It contains 3 phosphate. So this is a phosphorylated nucleotide. ATP is also a nucleotide, but this is a phosphorylated nucleotide. As we have discussed earlier, nucleotide is what? This is a combination of pentose sugar. Here in ATP, the pentose sugar is ribose, and a nitrogen base is attached with ribose, making it a nucleoside, which is adenosine. But when phosphate is added, it becomes nucleotide. Okay, so in ATP there are 3 nucleotides. That is why 3 phosphates. That is why this is called adenosine triphosphate. Simply when ribose is attached to adenine, this is called adenosine. When this adenosine is attached with a phosphate, it becomes a nucleotide, and this is called adenosine monophosphate. When it is attached, this adenosine monophosphate is attached with another phosphate, this is called adenosine diphosphate. And when it is attached with another phosphate, this is 3 phosphates are added, then this is called what? Adenosine.
Divorce.
That's all for today.